Hi everybody and welcome to our module on flow cytometry. Flow cytometry is a laboratory technique, and in order to understand this technique it's helpful to know where it gets its name from. So flow is the motion of fluid and cytometry means the measurement of cells. So flow cytometry is an analysis of cells as they flow in a liquid through a narrow stream. And a key point here, and the one that you want to remember for step one of your boards, is that this is a lab technique to analyze cells. We can analyze cells by size, by granularity, by their surface proteins, but this isn't used to study antibodies or antigens or DNA. This is used to study cells. Shown on the screen is a picture of a flow cytometer. The key components are a flow cell, which moves cells through the machine. There is a laser, which bombards the cells with light that will be scattered, and then there is a photodetector, which detects the light that is scattered by the cells. Here's a picture you can keep in your mind just to better appreciate this. We have cells flowing in a liquid stream. There is a light source which is bombarding the cells with light. The light is being reflected and the photodetector is picking up the light. And based on the characteristics of the reflected light, the photodetector will characterize the cells. It turns out that a flow cytometer creates side scatter and forward scatter. The physics of this is very complicated, and I don't recommend you try to learn it for the step one exam unless you have a special interest. But for our purposes here, just know that the forward scatter of light is based on the size of the cells, and the side scatter is based on the granularity of those cells. What this means is that you can pass different populations of cells through the flow cytometer and plot the front scatter versus the side scatter of the different cell types. And if you do this, you will get a classic graph like the one I've shown on the screen here. This is output from a flow cytometer, and there are a group of cells that are relatively small, as indicated by the small amount of front scatter, and they have very little granularity. Those are the lymphocytes. The granulocytes, on the other hand, are relatively big, so they have lots of front scatter. They're also very granular, and they have lots of side scatter. And then monocytes are in the middle. And this is a classic flow cytometer output graph. Flow cytometry becomes an even more powerful technique when it is combined with antibody staining. This process uses specific antibodies that bind to surface or intracellular proteins on cells being studied with the flow cytometer. The specific antibodies are tagged with a unique fluorochrome that reflects a particular wavelength of light. The flow cytometer will detect the fluorochrome if it is bound, and if it is bound to the cells, it indicates the presence of a particular protein in those cells. You can do this with multiple antibodies, each one with a different fluorochrome attached to it that reflects a different wavelength of light. In this manner, you can detect multiple different proteins in cells with a single cycle through the flow cytometer. So for example, you could use one fluorescent antibody that looks for binding to CD8 in cells. You could use another one that looks for binding to CD4 in cells, and then you could run populations of cells through the flow cytometer. If you look at the output shown on this graph, what we've got in this population right here is a collection of cells that is generating very little fluorescence along the x-axis, which indicates binding to CD8. It's also generating very little fluorescence on the y-axis, which indicates binding to CD4. Therefore, what we've got here is a population of cells that is CD4 and CD8 negative. In the top right corner, in contrast, we have a group of cells that is showing lots of fluorescence for CD8 along the x-axis, and also lots of fluorescence of CD4 as indicated by the y-axis. Therefore, this group of cells is CD4 and CD8 positive. Over here we have a group of cells which is generating lots of CD4 fluorescence but not much for CD8. So this group must be CD4 positive and CD8 negative. And then finally over here we have a group of cells generating lots of fluorescence with the CD8 antibody, so it must be CD8 positive, but it's negative for CD4 and therefore generating very little fluorescence on the y-axis, which indicates CD4 positivity. One of the clinical uses of flow cytometry is to detect fetal maternal hemorrhage. This occurs when fetal red blood cells cross the placenta and enter the maternal circulation. It's seen in cases of placental failure or trauma. It can present as decreased fetal movement or abnormal fetal heart rate, and it can cause a stillbirth. Flow cytometry can be used by employing monoclonal antibodies to hemoglobin F. If you detect significant amounts of fetal hemoglobin in red cells in the maternal circulation, it indicates that fetal red blood cells have crossed over to the maternal side of the placenta. Another clinical use of flow cytometry is in the diagnosis of the rare condition paroxysmal nocturnal hemoglobinuria. I discussed this condition in detail in the immunology video on complement because in this disorder, red blood cells inappropriately bind complement and lyse. But for our purposes here, know that you can diagnose it with flow cytometry by using fluorescently labeled monoclonal antibodies that bind to GPI anchored proteins. These are the proteins that are deficient on the red cells of patients with PNH. They include proteins like decay accelerating factor, which is DAF or CD55, also MAC inhibitory protein or CD59. And what you will see via flow cytometry is that the red blood cells in a patient with PNH will show reduced or absent binding of the monoclonal antibodies to these proteins because in this condition those proteins are absent. And that concludes our video on flow cytometry.